Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator here at Simulations Plus. I'll be hosting today's webinar, Behind the Scenes, making sure we don't have any technical difficulties. In this webinar, presenters Lisl, Shoda, and Steve Chang will explain the value of QSP in drug development, as well as common and innovative applications. They'll share case studies of how the use of QSP has influenced decision-making and program direction in a variety of therapeutic areas. A few housekeeping items before we begin. We take your privacy rights seriously. By registering and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. Before we begin, I'd like to conduct a short poll to understand your current familiarity with QSP. I'll go ahead and launch that now. And the question is, how involved are you with QSP? And I'll give everybody a few seconds here. Okay. Thank you, everyone, for your responses. And now I'll hand it over to our moderator, Nate Muser, Director of Alliance Management, to introduce our speakers.
Perfect. Thanks, Jasmine. I'm excited to introduce Lisl, Shoda, and Steve Chang, two amazing leaders in the QSP modeling space. Lisl is the Associate Vice President and Director of Immunology within our QSP business unit, with a focus on predicting and improving the understanding of drug actions on innate and adaptive immune responses, specifically representing adaptive immune responses in the liver and their contributions to drug-induced liver injury. Since 2002, Steve was the president and CEO of Immunetrics, a modeling and simulation company recently acquired by Simulations Plus. With more than 3 decades of experience in identifying emerging market needs and combining state-of-the-art technologies and resources to meet those needs, Steve continues to drive QSP improvement and bring new capabilities to QSP within Simulations Plus. So without further ado, I'll hand it to Lisl and Steve.
Great, thanks, Nate. Let's go ahead and get started. Okay, so today we're going to start with a brief introduction to Simulations Plus and to QSP particularly. We're going to walk through a few case studies that sort of illustrate some of the ways in which we have impacted clinical programs. And then we'll shift gears a little bit, talk about developing a standard for QSP at Simulations Plus, and then do a deeper dive into our capabilities in the particular area of multiple myeloma and how that can support clinical development. Okay, so Simulations Plus, we're a global modeling and simulations company. We have products and services in multiple areas, including cheminformatics, PBPK, QSP, ClinPharm, pharmacometrics, and regulatory strategies. And we operate in multiple different regions and really address multiple areas with a primary focus, or at least the bulk of our work, supporting drug development, drug discovery and development. I think for most people on this call, there'll be a lot of familiarity with some of the challenges that are faced in drug development. One of the key ones, I think, is failures occurring at the level of clinical trials. This figure on the right is from a relatively recent paper looking at the Pfizer experience. And what we can see is that, in terms of their success rate in clinical trials, in 2016, Pfizer in blue was below industry standard in gold, pretty much across the board. And then by 2020, Pfizer was well above industry average for clinical trial successes. And they attributed this to an increased focus on biology and on quantitative decision-making, which included modeling. So I think that there's, from the industry perspective, there's certainly evidence that modeling can contribute to better successes in drug development. Now, if we also consider the pharma perspective, the figure on the right is taken from a paper by Jane Bai at FDA, and illustrates the increasing number of QSP included in submissions to the FDA between 2013 and 2020. And on the left, we see sort of a timeline that lists some of the highlights for QSP at FDA particularly, but there's probably a parallel timeline that can be shown for milestones at other regulatory agencies as well. Starting from first FDA submissions, and then very recently in 2023, the FDA co-hosted a workshop on creating a roadmap for QSP-informed rare disease drug development. So I think a big a leap in terms of awareness and understanding of the potential value that QSP can bring to drug development. So I want to talk a little bit about QSP at Simulations Plus. We have, we've been doing this for a while, both within, as Nate alluded to, the Immunetrics group, but also at Simulations Plus. And I think there are a number of different areas where we have influenced programs. And so I'll just touch on these very briefly. Those of you familiar with QSP will recognize that trial design is sort of a sweet spot. And so there are a number, over 40 that we counted, large-scale clinical trials that have been informed by QSP. QSP represents the mechanism of action or toxicity for drugs. And so oftentimes as part of these projects, there's insights gained in terms of how the drug is working, what are some of the redundancies and ways in which different mechanisms come together, timing issues, etc. Because QSP represents heterogeneity in patients, there's the opportunity not only to look at response, but also non-response, and think about how that might influence patient selection criteria or potentially the identification of biomarkers. And then we've executed these projects across multiple different phases of clinical development. Some of these have been prospective predictions, which were followed by the actual clinical trials. And we have validation examples where the QSP project sort of predicted that modest improvement in the clinical trials, and that was subsequently borne out. Or cases where the QSP was predicting a success, and again, that was borne out. And finally, just like that last slide that was focused on FDA increasing experience with QSP as part of communications by sponsors, we've seen that as well. And in a few cases, we've certainly been able to put the learnings that are coming from QSP front and center in terms of potential impact on those regulatory decisions. Okay, so we've now talked about QSP as being increasingly recognized, QSP as having some concrete examples of influence on clinical programs. What is QSP? And that, the answer to that question is going to vary by the particular individual. But I think we could probably agree that it is mechanistic modeling, that it includes a systems perspective of health or disease, and that we're really looking at the interaction between biology, biochemistry, and the drug with respect to predicting safety and efficacy. There are multiple different types of models that can be put together, and that includes fit-for-purpose models that tend to be smaller in scope, narrowly focused to address a particular question, often created de novo and generated on a relatively rapid time frame to try and address that specific question. You can contrast that with platform models, which may take longer to develop, but have a broader scope of disease representation, allow the integration of multiple different targets or drugs, and are really intended to be reused for multiple applications, potentially following a program along. And then building on that platform model, there are disease progression models, where disease progression can refer to the progression of disease, where you have worsening over time and associated changes in some of the clinical endpoints, and or it can refer to the sequencing of treatments. So, from patients coming in with diagnosis to receiving a standard of care to perhaps having no response or inadequate disease control on that standard of care and moving to second line, third line, or experimental therapies. And so all of that is within the QSP landscape. I'm happy to say that Simulations Plus does include examples of these, all of these, although certainly we have more in the platform and disease progression models. So more specifically, what do we have? I'm not going to go through all of these in detail, but we have a large number of different therapeutic areas represented already and ready to go. So these include, on the top left, inflammatory and fibrotic disease, such as non-alcoholic fatty liver disease, NAFLD or NASH. We have QSP immuno-oncology applications, including acute myeloid leukemia and multiple myeloma, which we'll talk about in more detail in a few slides. QSP and autoimmune diseases, rheumatoid arthritis, SLE, IBD, so a number of areas there. And then in the safety space, drug-induced liver injury and drug-induced acute kidney injury. In addition to sort of having these models, I think one thing to point out is that we have the scientific / modeling staff that are well-versed in these therapeutic areas and can partner with programs to really bring, not just the modeling, but the mechanistic insight, the disease insight, and hopefully, advance those programs at a rate that is accelerated relative to what they would do in the absence of QSP modeling. And finally, one last point, which is that either through internal R&D or in collaboration with external parties, we continue to build on this library and develop additional therapeutic areas. And so if you were to check back in a few months, you might see that this list has grown. Okay. With that very brief introduction to QSP, I do want to go ahead and jump into case studies. We'll start with imvotastat, which was initially trialed for solid tumors and then terminated when 2 patients experienced drug-induced liver failure. So, after that, there was the proposal that AML might be an area where you could get efficacy with lower dosing. But of course, you have this experience, right, where patients have gotten injured, and you certainly don't want to repeat that experience. And so the question is, will those lower dosing regimens be safe for the liver? And so the decision was made to apply DILIsym, one of our safety QSP platforms, QST, I guess in this case, to look at liver safety and predict retrospectively what was observed in the clinic originally, and then prospectively what one might expect for these lower doses. And if you look, this work was presented by Kyung-hee Yang at the ASCPT earlier this year. But if you look at the right, the inputs for this are really the exposure to imvotastat, as well as an understanding of mechanisms of toxicity based on in vitro data. And to incorporate this in our QST platform DILIsym in order to make those retrospective and prospective predictions. And if we then sort of cut to the chase in terms of what we're finding, the historic protocols in which there were clinical observations of liver injury did reproduce dose-dependent hepatotoxicity. For those of you that are less familiar with the evaluation of drug-induced severe hepatotoxicity, or EDISH plots, these plots show peak ALT on the x-axis and peak total bilirubin on the y-axis. In this case, if we're looking at the far left figure, all of these simulated individuals, almost all of the simulated individuals, fall within the normal range. One person here with experienced an ALT elevation. As you increase the dose, more simulated individuals see elevations in ALT, and some actually move into this what is referred to as Hy's Law range, where there are elevations in both ALT and total bilirubin, and are at risk for severe liver injury. And we can contrast that with the lower doses, so 40 mg, 20 mg BID for actually a longer duration, 32 weeks, as well as a slightly higher dosing regimen, but still much lower than what was used originally. And what we see is now all of the simulated individuals fall into the normal range, suggesting that that one could take imvotastat forward with a lower dosing range and a reasonable expectation of safety. And of course, there's the mechanistic background behind that as well. So, and in fact, this has borne out in clinical trials where it has been taken forward into clinical trials. Thus far, everything is looking good with respect to minimal liver signals and relatively safe. So I think this is a nice example of being able to use QSP to evaluate planned clinical protocols, dosing regimens, and sort of increase confidence, as well as providing a mechanistic backdrop for why the rationale for going forward with those particular doses. One thing we didn't get a chance to do too much in that particular example was talk about the mechanism. So I'm going to switch gears slightly and talk about one other case study, which is on FGF21 analogs. So there are a number of these that are in development to treat metabolic diseases. And thus far, there's been variable efficacy reported for clinical trials. And so there was an interest in representing these FGF21 analogs in a QSP model of non-alcoholic fatty liver disease, NAFLD-SIM, and there's sort of a schematic shown on the right that gives you a high-level sense for what is contained within NAFLD-SIM, and to be able to really query this QSP model with respect to the mechanisms driving the FGF21 analog effects and how that might relate then to clinical endpoints. One of the great opportunities of working on this type of project is the chance for our scientists and program scientists to work together in terms of building out our understanding of what is happening with a particular compound and representing that mathematically within the models. And so in this case, we can sort of dive into the molecular details. And we have FGF21 on the right that is interacting with the FGFR1 beta-klotho receptor and inducing production of adiponectin. And then multiple downstream effects of adiponectin that can subsequently interact and influence the pathophysiology in the liver. And so if we then carry that forward and say, okay, we're going to do these head-to-head comparisons for these 4 FGF21 analogs. We're going to account for differences in compound exposure, as well as potency in these different analogs. And then we're going to select simulated patients that are aligned with the clinical data. One of the things that we can do is to look at and sort of build confidence in your mechanistic understanding, and are you reproducing what available data there are in terms of clinical intermediate clinical endpoints, if you will. And so in this case, data were gathered from multiple different sources on increasing adiponectin in the presence of FGF21 analogs and decreasing liver fat. And so if you, there are multiple different compounds represented here. In the colors are the clinical data pulled from multiple sources, and in the red symbols are the simulation results. And so what we can see is that there's a wide range of increase in adiponectin that's been seen across these different compounds, and correspondingly a wide range of reduction in liver fat that was recapitulated in the simulations. So that sort of gives sort of that looking at an intermediate endpoint and building confidence that yes, the representation is reasonably aligned with what we expect from the clinic. You can then take that to predictions of clinical endpoints, including something like fibrosis stage. And so not all of the endpoints that are predicted are shown here for the purposes of legibility, but what I do want to illustrate is that, we can look at multiple different compounds shown here, different protocols, and in different patients. So we're looking at patients with NAFLD, but F3 fibrosis or F4 fibrosis. And then we can look at the corresponding effect on adiponectin, on liver fat, and fibrosis stage. And you can do these sort of in a head-to-head way. In this case, we elected to demonstrate the results as a heat map for purposes of visual comparison. If, for example, we take this bottom one, Bio-89-100, and we look at, green indicates that it's one of the stronger compounds / protocols at inducing adiponectin. And as well, it's one of the stronger predicted compounds that influences fibrosis stage. And while not all of the data have been publicly disclosed in exactly the same way, I would say generally for these various compounds that the available data that's coming out in the media has suggested, sort of a corresponding relative efficacy across these different compounds. So hopefully this gives you kind of an idea about where, mechanistically QSP can sort of bring a better understanding to the team in terms of building, representing particular pathways, influencing particular pathways, building confidence by looking at intermediate endpoints, and ultimately predicting clinical endpoints. So with that, I'd like to have us switch gears, and I'll turn the presentation over to Steve.
Thank you, Lisl. If we look a little bit at the broad industry called QSP, a lot of the players will agree that it needs some level of standardization. From different perspectives, regulatory agencies see, and because they're seeing this increasing number of submissions with QSP, they're all bespoke right now, built for purpose models, using a diversity of different languages for implementation, using different modeling platforms and development systems, different techniques, even different methods of representing the mathematics for QSP models. And their ability to assess and understand the models within the briefing document response window, which is typically 30 to 60 days, is a difficult challenge. Pharma, including QSP in their submissions, are also sort of left to make educated guesses as to what would be required by the regulatory agencies in the midst of there being general guidelines, but not necessarily specific guidelines. And so they're also left guessing, how much do we show? How much do we give? Do we give all of it? If we do, that's a lot. How do we allow them to understand and digest it in a timely manner? The standardization effort is not a new theme amongst the industry. Their previous efforts have been largely unsuccessful. Those typically were to gather a bunch of the large pharma together in a room, have a consortium, have discussions about what is the right standard for such things, lots of discussions, but not really any substantive progress over the last 5 to 10 years. Next slide. SLP is beginning to introduce QSP standard. It is SLP's intention to have a QSP standard available for its customers. This would encompass all aspects of QSP, including how models are implemented and extended, includes how do you handle and create virtual populations, how do you fit those virtual populations in a lot of the settings to all relevant phase 2 and phase 3 drug trials in a particular disease. Consistent approach to model fitting and validation. So a lot of things that right now are done across a lot of different methodologies, a lot of different ways. We are looking to push a standard across a library of the models. Within these standards, broader clinical communities can be then educated on what these models can do, how to trust the model outputs, and that goes from a 1,000 front battleground right now as it is, to a more unified front. So that should become a little bit easier for everyone. And these levels of standardization would make it easier on both sides, regulatory and pharma, to operate within a familiar environment. Next slide. Here we'll sort of transition into the overview of the current license-ready multiple myeloma model. The model represents the critical biological elements of multiple myeloma disease biology and drug mechanisms of action. A lot of the core players that you would expect to be there are represented in the model, which includes various aspects of the disease biology, including biomarkers, immune cells, and cytokines. The model generates clinical biomarkers and cell counts in order to then generate clinical endpoints. In this model, and this grows over time, but there are over 30 phase 2, phase 3 clinical trials that are used to train this model. They span over 25 drug regimens and 14 distinct therapeutic targets. Drug MOAs include proteasome inhibitors, IMiDs, CELMoDs, dexamethasone, monoclonal antibodies, cell engagers, CAR T-cell therapies. Fitting the same disease model to as many therapies simultaneously adds another layer of helping to calibrate the core disease biology and specific drug MOAs to harmonize everything into one picture of the disease. Being able to also connect this to clinical endpoints gives you the visibility that most clinical programs need to be able to inform their decisions, make design changes, and inform their trials accordingly. The resulting group of virtual patients that is produced by this model is one that is representative of the real multiple myeloma patients that are likely to be enrolled in actual trials. So what this also does is it adds a layer that as you predict whether it's efficacy and whatnot in your new target or an existing combination therapy that you're looking to evaluate, or even something that combines an existing therapy with a novel one, your ability to then predict what that performance of that real trial would be is made available here. Next slide. We also have at SLP a standard for assessing model fitness, or how good is the model's fit to the actual data that we're fitting to, as well as how good does the model reproduce validation data that's been withheld from the model to test the quality of the model and its predictive nature. So in addition to a score that is used during optimization, something that probably is familiar to those of you who use objective functions, we employ an alternative measure of model quality that we term coverage that is independent of the training process. Coverage is defined as the percentage of data within the model's 90% confidence interval. The confidence interval is our model's estimate of the range of trial means that would be repeated trials would yield. So we're trying to get a mean behavior out of a population and see if that falls into the confidence interval for the model. By definition, we expect 90% of the data should be inside 90% of the confidence interval, not 100%. And more practically speaking, a QSP model that performs well is likely closer to a 70% coverage due to various factors, including, but not limited to, subtle differences in trial populations used for training, differences in how clinicians measure disease, especially diseases for standards of care that are rapidly evolving, other sources of noise, and just real-world variability. Coverage can also be assessed across virtual populations and models, and is thus a useful comparator for ever-evolving models, from one fit run to another fit run, or for one version of the model to another version of the model. This becomes a useful metric. To give you a snapshot of overall quality of fit, to the right is a parity plot that shows and highlights the quality of the fit across all response endpoints in this license-ready version of the multiple myeloma model. On the left-hand side, you see the actual value that the model is trying to fit to. The fit dots are in blue, the validation dots are in orange. Along the bottom are the model's corresponding mean value for each of those actual mean values that you're trying to fit to. And obviously, if there was 100% concordance, perfect data, perfect model, you would see that dotted line full of dots. In this case, you see quite a healthy spread of dots along that axis that shows as we are simultaneously trying to fit 500, in this case, 569 clinical trial mean values simultaneously across those 30 trials that I mentioned earlier. We are doing that in a virtual population that then subsequently validates to the 194 clinical trial mean values quite well. Now that you have such a model, what are some of the things that you can do with it? We'll show you a few prediction examples, particularly of combination therapies, since that seems to be of high interest in the multiple myeloma drug development space right now. What you see here is a graph of multiple things. So this is a slightly busy graph. You have PR for partial response, VGPR for very good partial responders, and then CR for complete response. And what you see here is a bar chart of the patients that respond in the model is in the actual solid colors by the actual data. In this case, published data is in the dotted, the lines that are on a diagonal. And what you see here, very good concordance between monotherapies, which the model was trained to. So that's DARA and TAL at a couple of different doses using a couple of different protocols. And then the model then predicts what would the combination therapy of combining these two drugs in a single protocol, in a single trial, result in terms of performance and overall efficacy of the drug combination. And you see here, the model does quite a reasonable job. Well within the model's confidence intervals of 90%, which is the blue bar, the thin blue bar that you see along each of the model lines, of which there are 5. And you see here that there's good concordance between the actual data and the model to results that it has not been trained to. As we try to quantify that further, as we try to quantify that further with the confidence interval comment that we mentioned earlier, for DARA, all doses, there's a 93.3% concordance. There's 100% concordance with TAL, and then the TAL plus DARA prediction has 75% concordance across all doses for all the data points that were available. Okay. The model is also able to predict DARA plus BORT combinations. Same style of chart, obviously slightly different drugs. Here we have BORT and DARA in monotherapy. Again, you see reasonable fit to that data. And then to the right, you see the prediction blinded of the combination therapies themselves. Same style where you have PR, VGPR, and CR for this. The training data, the model, and the trials themselves are listed on the right. And again, confidence intervals for the fit and then predictions are 100% coverage for BORT, 93.3% for all the doses of DARA. BORT plus DEX has 63%, and DARA, BORT, and DEX, triple combo, has an 83.3% concordance with reality based on an untrained setting. The model is also capable of predicting multiple clinical endpoints. DOR is another example of that endpoint. So the durability of response is an important indicator, especially in pretty much all of the different cancer indications at the moment, right? So the model training included DOR for the BORT monotherapy, And what we're doing here is predicting DOR for the combinations themselves. Here we have good fit performance for the model where we get 100% Bort mono, and for both Bort Dex combination, 47%, so it didn't do as well. But for the Darabort Dex combination, triple combo, it did extremely well. And back to the chart on the left, you see the percent of responders, and over time, how long they continue to be responders. What you see there is in the solid lines is the model prediction of each of these different protocols. The X's are the actual published data for those different protocols over time. And you see here that you get quite good concordance, especially in the red triple combo case. So if you're interested in how you might be able to enter into QSP with Simulations Plus, we have a variety of ways of doing this. This is just sort of a sampling of them, but we're generally flexible and can accommodate. We have consulting projects with fixed price based on milestone payments, time and materials with monthly payments. We can even do licensing, so it's available for in-house use. We have high performance grid licensing available as well. We can include some training for that. From an FTE standpoint, we can dedicate scientists to your projects, and then a consultant coach arrangement where we do a consulting project culminating in a fall delivery and a short license for some of our models. And we facilitate ongoing queries by sponsors. If you're interested in learning more, we have multiple ways of trying to reach us. Feel free to reach out to Lisl or myself directly, or feel free to reach out to any of the Simulations Plus people that are here online. Thank you for your attention.
Thank you, Steve. So before I pass it off to the Q&A, I do have one last poll I'd like to launch. And this one has this one has 2 questions, and I'll give everybody a few seconds to answer those. Okay, I do see some answers coming in, so I'll give it a couple more seconds. Okay, thank you. I'll now pass it off to Nate for some Q&A.
All right, thanks, Jasmine. There are a couple questions in there. I think one of them is around regulatory agencies. So, has any part of the platform been qualified by a regulatory agency, and/or how do regulators typically, respond to submissions? So maybe you guys can speak to the regulatory piece of QSP.
So I can go ahead and kick us off. One of the examples that we were talking about was QST and DILIsym in particular. And so, that DILIsym has its origins in an initiative that's a public-private consortium to build this model. And the FDA was sort of part of that conversation from the beginning, not necessarily influencing what was being built, but certainly aware of the effort and interested in where that went. FDA has licenses to DILIsym and has continued to do that over many years now. In terms of exactly, what in terms of our interactions with the FDA, I would say it's sort of a mixed set depending on the particulars. There are times when the sponsor sort of takes the results and, independently decides how they're going to present them or use them in the context of the other materials that they're presenting. There have been occasions where we were part of the preparation, say for an ad board, to really understand what was happening with a particular case. And, there have been times when we were part of putting together briefing docs or presentation materials. With respect to the big question about qualification, I think this really goes back to the heart of what Steve was talking about sort of midway through the webinar, which is that, and you could even see it in one of those intro slides when we were talking about the industry interaction with the FDA in particular with respect to sort of setting standards. And I think that what has emerged from that is that, we have as a community, I think we have some things that we point to as important. Is the, what question is being asked, using this modeling and simulation, and what is included in the platform? How do you qualify that the simulations are giving you something that is reasonable relative to reality? Is there a set, either you call it training and testing or calibration and validation, or, et cetera, et cetera. How do you capture heterogeneity in responses? There are many things I think that we sort of agree that are important. Building something that is like a guidance for all is, I think, tricky, right? Because there are so many different types of QSP models, and therapeutic areas differ. One therapeutic area may have hundreds of trials, and another therapeutic area might have a handful. And so, it's really, I think, tricky to put together sort of a one size fits all. That said, I do feel like the community is still moving in the direction of trying to figure out what could we put together as a framework that would be, sort of a guidance for, that applies to multiple instances. And I think, Steve, if you want to talk about that, that's exactly sort of the conversations that we're having internally to try and figure out, what could that SLP standard look like?
Absolutely. So we've participated in a number of briefing documents as well, and almost to the point where we sort of have a standard outline for what we include. There is typically full disclosure of the model, the construct, the biological assumptions, the parameter settings, et cetera. But it's so much stuff that it's typically like 80 to over 100 pages. And so it adds quite a bit of volume to the submission. When we've had discussions with the FDA, not specifically about an individual submission from a sponsor, but just in general, because they're trying to wrap their head around QSP, one of the things they always refer back to, and I think you've heard it in other settings with the FDA too, they look at the overall body of evidence, right? And they also weigh, if you're trying to use QSP to justify a certain action by the agency, they weigh the body of evidence relative to the action that you're trying to ask for. So obviously, to give you a couple of examples, if you have a recommended phase 2 dose, you use QSP and CPP to sort of back up why your decision for the phase 2 dose was X. But then you're going to go ahead and test the phase 2 dose. That bar is a little bit easier for the FDA to accept. On the other hand, you can go to the other end of the spectrum. You have a rare disease, you have a drug that passes safety, and now you want to jump straight to a phase 3 trial with like 2 or 3 patients, and you're going to justify everything with model-informed drug design, MIDD. They're going to have to take a very hard look at that, right? That historical evidence and everything else to be able to say, can we really trust what we have here based on, especially in an environment where it's limited data, right? The FDA, with respect to rare diseases, they realize how difficult it is to do rare diseases, that they even have this saying that they use at the rare disease meeting, a trial of N equals 1. Can you imagine the FDA approving a drug for a rare disease based on a single data point from a single human, right? That's something that's a challenge, right? So how do you back up that data with a lot of other things is definitely something that the agency is trying to wrap their head around. But it's an ongoing process for sure, like Lisl said.
Okay, thank you guys. So a few questions coming in around, I think the actual building and potential validation of a QSP model. So, what are the stages of building a reliable model? And how do you go about that build as well as, validating the model as well?
Think there's a lot of philosophies around it. I think one of the philosophies that we use at Simulations Plus, so there's a lot of key aspects of it, right? Understand the biology you want to represent mechanistically, gather the data that's available to help you constrain the dynamics of the model. Those are probably the two key linchpins, because absent good biology and assumptions, and absent some data, right? Some people say you don't need any data for QSP models. I would kindly like to disagree, because if you have no data, you have basically one of these diagrams without any quantities of anything, right? So you do need both of those. Some people do that iteratively, others do that differently. So there's variations on doing that. And I think when it comes to both fitting your model to data and validating your model to data, it does it can be very specific to what data that you have available. So for example, in the model that we reviewed today for multiple myeloma, in that setting where you have the ability or have at your disposal 30 distinct clinical trials to be able to decide which ones fall into fitting, which ones fall into validation, it's much easier and more straightforward to say, okay, why don't we fit to these drugs and these trials, and then validate to these other drugs and trials to sort of test the model. One of the most important aspects of validation for a model is you want to try to confirm that you're not what we call overfitting the model to your actual data, right? So what you can do is my model fits my data perfectly. It's great. Let me go predict something. Ah, crap, that's not even remotely close. What did I do wrong? I fit my data perfectly. And that was part of your problem. You fit your data so well, the only thing your model could tell you is what you told it, right? And that's when models are scientifically maybe interesting, but they're really useful if they can tell you something that you don't yet know. And that is honestly an art still in QSP, right? Those who do it well, do well. Those who don't do well, we don't have to talk about those.
Yeah, I guess I would just add that there have been multiple publications in the literature sort of talking about the process that you take for building a model. And I would say that, we broadly take a very similar process to what's been published out there. One thing that I think is valuable to think about with respect to these mechanistic models is that of course, you're going to try and use all the available clinical data to constrain your model and to make sure that it is reproducing reality as we have tested it. But I'd say we also have the ability to sort of interrogate our models with essentially sanity checks. So you can, if you have 10 different mediators represented in your model, and there are clinical data out there that are targeting 3 or 4 of them. Well, okay, you're pretty confident in those, right? And you can, inhibit TNF alpha and see what happens, and be, and there are multiple different ways you could do that. But maybe you have, several others that are less, for which there are not clinical data, but there's sufficient data in the literature to tell you that this could play a role, and that it is maybe a critical redundancy in some patients that you want to capture. You can still go in there and block that particular molecule and see what happens, and be sure that your model is behaving in a way that is reasonable. We have different terminologies for how we characterize these things, but in essence, we are, because it is a mechanistic model, you have the ability to do all the what if experiments. And this also gets to maybe the importance of looking at particular therapies. So some therapies, if we're representing mechanisms of action, you could be very clear on some of the mechanisms, but have hypotheses around the others. And you can do the parallel experiments, right? What if I include hypothesis X? What if I don't include it? How does that, influence my predicted outcome, and how does that help me to understand better what experiment I could do to figure out which of those two scenarios is true.
All right, thank you guys. The next question is kind of around context of use. So it says, when might you consider DILIsym for qualification, specifically from a non-clinical perspective? How can DILIsym help to predict safety endpoints?
So we're talking about sort of early use case for DILIsym. Yeah. So that's great question, and something that we're very interested in getting into. So the scenario, and we have a few members of that public-private consortium that I talked about that have sort of looked at this in greater detail. So part of the challenge is that we are taking you have the potential to do sort of a Cadillac version of a liver safety analysis, right? Where you do a full PBPK model and you do a very detailed multiple replicates, multiple doses, in vitro characterization of their potential interaction with different mechanisms of toxicity. You put it all together, yada, yada. It's in the non-clinical space. A lot of times you want to look at multiple different compounds, and you want to do it quickly and inexpensively. And so, without going into the gory details, I would say that we have put together sort of a plan for how you could do that. And it could be, to do it much more quickly, much faster, and be able to say, relatively speaking, yes, no, across the different, across these, 10 different compounds that we want to very quickly be able to assess. So that's something that we could certainly follow up and have a deeper conversation on. That type of methodology has been sort of vetted with real data. And so I think that there are certainly opportunities to apply it in an earlier stage and to get to some of those quick safety questions on a good timeline.
Okay. This is sort of two questions that I think kind of go together, right? In QSP modeling, it says there are too many parameters to estimate. How do you deal with them? And are there methods to validate the model other than fitting? So maybe talking about the numerous parameters, how you deal with that, as well as any potential other validations besides fitting data.
Boy, those are rolling up your sleeves type questions. So I think that there are a lot of people who call QSP modelers is a different art form that's not specifically appreciated yet until you get into it and you start to face these things. I think some people's approach includes sort of incrementally setting parameters. Obviously, your best bet is if you have data to inform the value of a parameter, you want to use that. That's typically data that will be from publications, maybe cell half-lives and other things of that nature are generally fairly available. So those types of information can be locked down so you can reduce the number of parameters. I'll sort of touch base on this because this is probably where it ultimately leads to. Do you build entirely identifiable QSP models versus non-identifiable QSP models? What that means is that you have visibility into every parameter and what the range of values that parameter should have in your model. And there is sort of a branching point that ends up occurring within those who approach QSP. There are groups that entirely wish to stay within the identifiable realm. One of the challenges with that is it's very difficult to bridge all the different gaps that you need to bridge in order to get beyond something that might be a PBPK model, even a sophisticated PBPK model, to be able to remain identifiable. If you want to go beyond that and get into patient populations, etc. You can do that through a POP-PK approach with even a PBPK model, but it'll be slightly different than a QSP model. QSP models try and aspire to encompass more of the biology. And by doing so, they do need to make a decision traditionally of branching into that wild unknown, which is parts of my model are going to be unidentifiable. How do I solve that problem? At Simulations Plus, we've been wrestling with that problem for probably a quarter of a century in terms of calendar years, let alone person years, right? So we've got a ton of experience wrestling with that. And I can tell you, we do have solutions that work quite well, as proven by our track record and our resilience in the marketplace.
All right. I want to get this one last one in, be conscious of time here, but in your estimation, so what would be the percentage of time or cost savings to a molecule development plan that where QSP is implemented versus where it's not, right? So what are the benefits there from your perspective?
I could give you a couple of big ticket items. So in one particular case where there was only first in human data available, so not even any data in an actual disease, we were able to show that the doses that they were selecting in phase 2, where you traditionally want to be able to show an ascending dose response sensitivity to your patients and the drug, and you also want to be able to show the saturation level where your drug's efficacy tapers off. If you chose those doses incorrectly, you ran your phase 2 and you didn't show enough to the FDA, you're running another phase 2. In this particular collaboration, they actually trusted the QSP model enough. They changed their trial design to include some of the doses that they weren't originally considering because they thought they had it covered. And true enough, as they completed that trial, they were very thankful they did, because you know what the cost of a second phase 2 trial is. I can tell you it's nowhere near the cost of what the QSP work was, by more than an order of magnitude. Another setting was in a phase 3 trial where the regulatory agency was requesting for another dose to be added to the phase 3 trial. So you can imagine adding a dose to a phase 3 size trial. That's a 7-8 figure kind of number when you're adding an additional dose in this particular market. And so they went at they responded to the agency with both a QSP model response as well as the standard CPP response. And they said very happily coming back that lo and behold, the agency finally accepted and we didn't have to run that extra dose. But gosh, we've done this many times with just a standard CPP package, and they've always needed us to run the second dose, that extra dose. So just a couple of snippets in time. Obviously not, it's a sample, not the truth, but certainly very important big bang benefits to using QSP.
Yeah, I mean, I might just add another example to that sort of is in a slightly different space, which is the FDA's fixed combination rule, where you have sort of you're co-developing or you're developing a drug that has two different, or more than two sometimes, different active entities. And so in order to carry forward with those, you need to demonstrate that each of those are independently efficacious, and you need them together in order to get something better than what is already available. And so demonstrating that type of thing clinically, I mean, even 2, right, if you consider different doses, but if you start talking about 3, 4, 5, the combinatorics just get really complicated and very expensive very quickly. So, and this is one of the areas where QSP is a sweet spot, that you can, represent mechanistically each of those different active entities and demonstrate, the combinatorics on a much shorter time scale. So, and that has been used to support not having to do clinical studies in support of this fixed combination rule with the FDA.
All right. Thank you both for the presentation and the Q&A. I will hand it back over to Jasmine to close us out.
Thank you. So I just want to say thank you to everyone. For any questions that we weren't able to answer live today, our BD team will reach out to you with answers following the webinar. We invite you to visit our website to learn more about QSP, QST capabilities. You can also follow us on our social media. I put those links in the chat. This webinar has been recorded. Slides and video will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thank you.
Thank you.